Hello everyone, I'm Sidhvita, a member of Krino Forensics. In this video, we are going to discuss a very sensitive yet scientifically important topic, the forensic perspective of menstrual blood. This topic is especially relevant in sexual assault investigations, where correct identification of biological fluid plays a crucial role in justice delivery. Before going into details, let me briefly outline what we are going to cover under this topic. First, we will start with a basic introduction to menstrual blood. Then, we will compare menstrual blood with peripheral blood. After that, we will discuss detection techniques used in forensic science. And finally, we will look at future directions and concluding points. Now, before focusing on menstrual blood, it is important to understand vaginal fluids as a whole. Vaginal fluids include a range of biological secretions such as vaginal epithelial cells, cervical mucus, and menstrual blood. Among this, menstrual blood is particularly important in forensic science cases, especially in sexual assault investigations. But why? Because it can help in determining the time of intercourse and in distinguishing consensual and non consensual contact. Menstrual blood, scientifically called menstrual fluid, it is a mixture of blood, cells, and tissues from the uterine lining, also known as endometrium. It is a shed during menstruation when pregnancy does not occur. This fluid is usually reddish brown, slightly darker than venous blood, and also contains vaginal secretions and cervical mucus. So, menstrual blood does not just a blood, it is a complex biological fluid. Now, let us clearly define what menstrual blood is. Menstrual blood is regular discharge of blood and mucosal tissue from the inner lining of the uterus through the vagina. It is composed of blood, vaginal secretions, and endometrial cells. When compared to regular blood, menstrual blood is darker in color and poorly oxygenated. Importantly, menstrual blood is considered a waste product as it contains dead and non functional tissues. It also has lower concentrations of iron, hemoglobin, white blood cells, and red blood cells compared to regular blood. Menstrual discharge usually lasts 2 to 7 days and occurs every month, with an average menstruation cycle of 28 days. Now, let us look at the similarities between menstrual and regular blood. Both menstrual blood and regular blood are biological fluids. They are red in color and contain hemoglobin, red blood cells, white blood cells, iron, and proteins. Both also have a similar pH, which is why simple chemical tests cannot easily differentiate them. Now, let us focus on the differences, which are extremely important in forensic investigations. Menstrual blood is discharged during menstruation, whereas regular blood circulates within the vascular system. Menstrual blood contains cervical mucus, vaginal secretions, and endometrial tissue. Regular blood, on the other hand, maintains RBC, WBC, platelets, and plasma in higher concentration. Another key difference is that regular blood is highly oxygenated, while the menstrual blood is not. Now, let us briefly understand peripheral blood. Peripheral blood originates from the circulatory system, including veins and arteries. It mainly composed of RBCs, WBCs, platelets, and plasma. It can appear brighter and dark red, clots quickly, and usually odorless. Under microscopy, only blood cells are absorbed. In forensics, peripheral blood is used to detect injury or confirm presence at a crime scene. Now, when we compare menstrual blood with peripheral blood, the difference becomes very clear. Menstrual blood originates from the uterus during menstruation. It contains blood, mucus, and endometrial tissue. It is dark red to brown, rarely clots due to natural anticoagulants, and it may have a noticeable odor. Microscopically, it shows blood cells, tissue fragments, and bacteria. From a forensic perspective, menstrual blood is especially important in sexual assault and menstrual identification cases. Now, this is very important to understand why is this differentiation necessary. Menstrual blood may be unrelated to criminal activity. If menstrual blood is misidentified as a peripheral blood, it can mislead investigations and affect legal outcome. Correct identification helps in supporting or disproving statements given by victim or suspects. So, accurate differentiation is essential for fair and scientific investigations. Now, let us move on to detection techniques used to identify menstrual blood. Modern forensic science uses both traditional and advanced methods. The first one is immunochromatographic assays. These tests detect human hemoglobin and D-dimer. D-dimer levels are elevated in menstrual blood due to endometrial tissue breakdown. They allow quick on-site screening. However, they are less reliable in degraded or mixed samples, and cross-reactivity may occur in conditions like thrombosis. Talking about traditional microscopic and chemical methods, tests such as benzodine and Castelmeyer were traditionally used. However, they lack sensitivity and specificity and are not effective on aged or contaminated stains. Talking about Raman spectroscopy, it identifies biochemical differences between blood types. It can also estimate the time since deposition and it is non destructive. However, it requires expensive equipment and expert interpretation. Now, genetic and molecular markers. This is one of the most promising areas. MicroRNA markers are stable and reliable, even in old or mixed samples. Gene expression profiling shows clear differences between menstrual and peripheral blood. However, these methods need validation under real forensic conditions. Next, we have biochemical and immunological differences. Menstrual blood contains elevated cytokines and inflammatory markers. These provide additional indicators for differentiation. Menstrual blood contains endometrial stem cells, which are not present in peripheral blood. Although mainly studied for therapy, it adds biological uniqueness useful in forensic identifications. Talking about the limitations of field tests, some field tests such as serratic PMB test have shown inconsistent results including false negatives. This highlights the need of standardization and validation. Then comes integrated approaches. Modern studies recommend multimodal approaches combining visual examination, immunological test, molecular techniques, and spectroscopic analysis. This increases accuracy and reliability in real case work. Now, let us look at future directions. Our future research should focus on multiomics integration, combining proteomics and transcriptomics, standardization of molecular panels, AI and machine learning for data interpretation, stem cell specific markers, low cost field friendly kits, large scale validation studies, and ethical consideration, especially in sexual assault cases. To conclude, a multimodal approach combining biochemical, cellular, and molecular markers enhance forensic reliability. New techniques like immunochromatographic assays, Raman spectroscopy, and gene expression profiling improve accuracy. Markers such as D-dimer, microRNA, and inflammatory biomarkers help in differentiation. However, challenges of sensitivity, specificity, and cross-reactivity still exist. Hence, further research, validation, and integration of methods are essential to strengthen forensic evidence and uphold justice. Thank you for listening. I hope this video helped you to understand the forensic importance of menstrual blood in clear and scientific manner. In case if you have any doubts or want us to explain any forensic topics, please comment below so we will cover them in future forensic videos. And the presentation document will be shared on our social media channels. You can access it directly from there. Stay tuned to learn with Krino Forensics to explore the world of forensic science. Thank you.